Hello and welcome to anticipatory briefing number 8 of the 2025 GESDA Summit. We are talking about fungal biology and the future of food security. Fungal infections in crops are accelerating due to climate change, threatening global food supply and ecosystem stability. Understanding how fungal pathogens emerge and spread is key to building more resilient agricultural systems. How will fungal biology influence food security and sustainability over the next 5, 10, and 25 years is going to be the topic of a 15 minutes presentation followed by 15 minutes of Q&A. Our speaker today is the chair of the food security at University of Exeter. Please help me welcome Sarah Gurr.
Okay, good afternoon, everyone, and thank you for the invitation to speak. I speak on behalf of CIFAR, which is the Canadian Institute for Advanced Research, with their fungal initiative. So imagine 3,000 years ago, you're Pandora, and you're about to open Pandora's box. Well, when she opened the box in ancient Greek mythology, she let out all the evils on the world. However, at the bottom of the box was the spirit of hope. I want to talk about the spirit of hope with regards to fungi and food security today. For today, we face a world with a burgeoning population estimated to grow to 10 billion by 2050. That means we need to generate another 10 to the power 15 calories per year to feed this new population. Against that, we have volatile political conditions, we have land usage change, urbanization, and loss of agricultural land. We also have migration and we have climate change. But I want to talk particularly about the impact of fungal pathogens on global food security. And the message is fungi are the most important agents of crop disease, and that's rather unlike the balance of human disease, where I think we're more transfixed by viral and bacterial diseases, although fungal diseases are important on humans. And they must be controlled, fungal diseases on crops, to ensure global food security. So I want to put it in context because in the discussions I've been having with quite a lot of people here, people say, well, why are fungi important? So fungi impact every aspect of our lives. Today you will breathe in, over the next 30 minutes, about 1,500 fungal spores. You'll also shed over that time 900,000 epidermal cells. So beware your neighbor with dandruff, because that will give you an extra fungal load. So fungi are, in my world, citizens of modern society. They are ethnically diverse and ancient, meaning that we know the names, or we have named about 150,000 different species of fungi, but there are probably about 2 billion different fungi or fungal classes or even fungi phyla in the world. So they're ancient. They emerged from animals about 1 billion years ago. They're migrants. They hitchhike lifts and trade and transport around the world. And they also vortex themselves, particularly during hurricanes, so dramatic weather. And they can be deposited thousands of miles away, as in Hurricane Ivan in 2004, when soybean rust moved from South America up to Northern America, and this year to Canada. They're known as recyclers in carbon and nitrogen, and they're also absolutely pivotal to the mycorrhizal nets that form around 90% of land plants. But they're manufacturing factory workers, for without them we wouldn't have any beta-lactam antibiotics such as penicillin and cephalosporin, neither would we have statins or this wonderful new drug for the treatment of multiple sclerosis. Fungi are also scholars and teachers. Unbelievably, work on fungi has earned 6 Nobel Prizes. Probably the most notable and relevant today is the discovery of the cell cycle by Sir Paul Nurse and his colleagues, and that's impacted on every aspect of human life and medicine. But fungi are also villains and rotters. They rot our houses, make our feet smell, but most of all, they are agents of crop disease. And today, despite mitigation, the most important agents of crop disease are the fungi. And mitigation practices include the deployment of inbred, so traditionally bred, disease resistance genes, and also the widespread spraying of fungicides. Despite that, we lose between 10 and 23% of our crop each year in the field to fungal diseases, and we lose another 10 to 20% post-harvest. And this is something we could do something about to preserve food security rather better. So fungi jeopardize global food security. They decimate our calorie crops. And if you look at the pictures here from the left to the right, you're looking at wheat and rice and maize. And those three crops cover 40% of global agricultural land. You've then got soybeans and potatoes. On the left, you've got wheat with wheat stem rust. In the middle, you've got magnaportha, so rice blast. Then you've got corn smut, soybean rust I mentioned earlier, and an organism that isn't really quite a fungus. It's an oomycete that caused the Irish potato famine in the 1850s on potatoes, which is still a massive problem today on solanaceous crops. So despite mitigation, so despite inbuilt disease resistance breeding, and also the widespread spraying of antifungals, we lose enough to feed 600 million to 1.4 billion people just due to these diseases alone. So fungi are really important agents of crop disease, and of the fungi, it is the rusts and the mildews that are the moving fastest and most problematic. And you can see a rust on the wheat on the top left-hand side. So some early modeling we did, which was statistical, showed that fungi are moving in concert with climate change. They're moving a depressing 7.8 kilometers per year to the global north. And I'll come back to that. So at the moment, unless we embrace some of these more modern technologies that I'm going to talk about, fungicides remain our frontline weapon in the fight against fungi. But there's a problem, and again I'll return to this, as there is dual use of quite a lot of the antifungals that we use, both on crops and in the clinic, and antifungal resistance has emerged, as I'll allude to in a minute. We also have a problem that the global pathogen burden is changing, and I'm going to show you, and I hope this runs. This is the burden today versus the burden in 2070. And these are mechanistic models that we've run in concert with the Met Office in the UK. They're based on vast amounts of data where the inputs are the host, so the crop, the pathogen, and climatic danger, the climatic conditions. So what they show is three things. Firstly, before you overlay the pathogens, we can show what sort of crops we can grow in the global north and in other parts of the world, particularly in the Sahara, that might nimbly adapt to climate change. So what this actually says is that the burden of food insecurity will increase transiently, but will restore as time goes by. What I mean by that is, sadly, there's going to be a lot less food in the Sahara and in Central Africa, and a lot more food in the global north. But after the honeymoon phase, when the new crops then become invaded by pests and pathogens that have moved, we'll go back to the same sort of food inequality that we have today. And you can see this illustrated by the changing months and the burden of more pathogens as time goes by. So what are we going to do about this? What are our societal and our scientific needs? So societal needs are really unbelievably important. We need to alert both politicians and the public alike to the plight of plants to disease. We all talk about human diseases, but if you went out into Geneva and you asked 1,000 people about disease, I doubt a single one of them would mention crop diseases. And if you're being extremely on the spectrum about it, you can say, well, if you don't get enough food, all the academic diseases of HIV, tuberculosis, even COVID, etc., just wouldn't happen. So this is really fundamental to food security. We also need to have succession. So somehow we need to make this a more popular subject so that the young folk follow us as we all fall off the mortal coil. Our scientific requirements are also manifest. Only can modeling and use of AI and everything happen if we are truly interdisciplinary. So for me, it's using maths and AI and as many large databases as is possible, but also informing these models with real data that we have taken from cell and molecular biology of these pathogens in the lab. So what's going to happen over 5, 10, and 25 years? And again, this is very difficult to divide it into finite years, so it's kind of a continuum. So you'll see a bit of a repeat on the slides as I go through, and I won't be able to talk about everything, so I'm going to pick a few stories as I go. So today, you're probably aware that thanks to the Green Revolution, we have monocultures. We have vast hectareages of genetically uniform crops, such as, for example, in Alberta in Canada, where there is one wheat field which is 15,000 hectares of genetically uniform wheat. Now, can you imagine if you were a rust fungus? You would see this as the most ideal feeding and breeding ground. And what you would do is go through a rapid life cycle with an incredibly plastic genome, and you would emerge resistance to overcome the disease resistance that's been built or bred into the wheat, and also to overcome the single target site fungicides which are sprayed. So, over the next 5 years, one of the most important things will be to use genomics in the rational design of fungicides which target features that we can show are truly fungal and which won't affect plants and won't affect animals, such as, for example, the fungal cell wall or components of mitochondrial respiration. We also need to test new fungicides in combination because, sadly, in our world, all of this is rather secret data being kept by the large ag chem companies, and it's not very fashionable to try and get students to do combination fungicide testing to look at the rate of emergence of resistance. So, fungicides remain our frontline weapon. And as I've said today, we rely pretty much on single target site fungicides. That means fungicides which target a particular process in fungi, such as the azoles, which go for the formation of the fungal membrane by affecting ergosterol biosynthesis. Because these are single target site chemistries, what happens is fungal resistance can emerge as quickly as 2 years after the introduction of a new fungicide. We've also got this problem, which has been highlighted quite a lot recently, and particularly by the medics, and particularly during COVID, which is as we've sprayed our crops with the fungicide azole, azole resistance has not only helped save the crop, but unfortunately what it's also done is it's transferred into Aspergillus growing on the soil. So Aspergillus fumigatus has acquired azole resistance, and now this means that this azole resistance has moved from crops to the clinic. And so the medics are having problems not prescribing azoles because they have azole-resistant aspergillus in the patient's lungs. And what we're trying to do is ring-fence drugs that can be used in the clinic versus those that can be used for crop sprays. So we're really reliant on drug discovery, and insights from the genome and AI will doubtless help us with this. So over 10 years, what are we going to embrace? Well, definitely disease prediction is going to help. Our tools are really very poor in plant disease epidemiological prediction. But we're also going to go towards looking at the mechanisms which underpin plant immunity, and also we're going to go towards gene-edited crops and the use of RNA crop trials. So let me try and explain some of these in a little more detail. So it might surprise you to know that plants have an immune system. They have both an innate and some form of an adaptive immune system. So what we can exploit is the fact that plants have an early warning surveillance of proteins which interact with the pathogen. So they recognize the fungal cell wall of chitin and glucan and turn on defense in plants. So we can immune boost plants, but we can also exploit disease resistance genes. So these are like a lock and key mechanism where a major resistance gene recognizes the avirulence component of a pathogen and it turns on disease resistance. That has been the basis of traditional plant breeding for disease resistance, and you can imagine has been overcome. But the more that we know about it, the more we can genetically modify by gene editing these features. And then I just wanted to mention one other rather glorious story from Denmark, where the plant breeders have collaborated with politicians, farmers, and everything, and they've come up with a series of cultivars which head at the same time in wheat. So they all grow to the same height, and they can be harvested at the same time. But they are mixed cultivars carrying different resistance genes, and they've been proven to be incredibly good at resisting fungal diseases in crops. So what about gene editing and RNAi? Well, the British government in 2023 embraced gene-edited crops to be deployed. And already around the world, there are a couple of quite interesting stories. One is of blackberries, which have been bred without the pips, not exactly relevant for food security, but a very clever piece of science. And another story where avocados and fruit have been edited so that the polyphenol oxidases in them are prevented from fruit going brown. But what realistically is there on the way to control crop diseases and therefore, hopefully, boost the number of calories to feed humanity? The answer is that we have crops which have been gene-edited to be resistant to Phytophthora, to the potato, and also gene-edited to prevent mildew infection in tomatoes. And there are lots of stories which are coming through, hopefully will be near market over the next few years. And we also have some beautiful stories, particularly from Hailing Jin's lab in Riverside in California, where they have used RNAi, so small molecules, which they're packaged in vesicles, and they're managing to spray them onto crops. So this circumvents the need for gene editing. So, very briefly, over 25 years, it will be the amalgamation of these absolutely brilliant technologies, hoping that the world also embraces gene-edited crops well. We obviously need a greater calorie diversity, so we need to use unusual forms to give us calories, such as pearl millet, lentils, etc. So we need to change our diet, because we're going to have to grow new crops in new places because of climate change. So hopefully, if we can integrate all of this, we might go to a policy known as LIFE, which is low-impact farming for the environment. So really, I just wanted to say that fungal diseases must be better controlled to ensure global food security. And the final outlook is really a point which is, if we go towards this kind of sustainable farming, we need far better investment in protecting our crops. And to do that, we need to raise awareness about the plight of plants. We also need a far stronger voice than we have, and hopefully we can tempt the WHO to make a list of pathogens which affect crops, as well as the pathogens that affect humans. And only then, I think, can we move towards a policy of One Health. Thank you very much.
Thank you, Sarah. Are we going to have questions from our audience? Please raise your hands, and we're going to bring you a microphone. We have one question here. Please state your name and ask your question. Thank you.
Thank you very much, Sarah. That was fantastic. And thank you for standing up for plant health, plant pathology. I want to ask you, I'm looking at biological control, and how do you see that fitting into that 5-10 year window?
Yes, I'm glad you asked. I didn't really have time to talk about it. So biological control is probably one of the more fashionable topics, but we're going to have to be really careful about biological control. So one of the fashionable topics at the moment is about using fungi that grow within plant tissues, the endophytes, and using those to control diseases. But what we must be careful of is that we don't make something that is immediately helpful into a pathogen, because the more we spread it out in nature and the more the inoculum potential goes up, the more likely we are to generate new pathogens. And there are a few stories that are beginning to happen like that. However, there's some also very exciting stories coming out from sequencing root biomes and seeing antagonistic and agonistic organisms in these. So there is potential, but I think it would only be part of a bigger practice of perhaps embracing gene editing, biological control, mixtures, and some antifungals which have been rationally designed. So I think we need an embracing policy. But thank you for the question.
Hi, I'm Juan Pedraza. I have a related question in terms of using phage type technology. So now that we can use AI to design specific phages for bacterial, for combating bacteria, do you see any sort of similar application for combating funguses?
Yes, there are some fungi which have phage on board. For example, the powdery mildews, and there's a project looking at the impact of control of mildew using phage technology. There's also an older story where in America, chestnut blight was controlled before phage really became that popular. And there's quite a nice story which needs to be revisited. So it's definitely an area that needs to be looked at.
Christoph Napels, Risk Insight. One big question I have for you, Sarah, is that are you worried that if we allow gene editing to come to market, right, and then you have that will generate a new monoculture product?
Yes, it's a real dichotomy. We unfortunately need monocultures if we're going to feed everybody, particularly if urbanization continues at the rate it is doing. And some countries in the world are literally concreting over agricultural lands and rivers. So I don't see there's an alternative to monocultures at the moment. If we go to gene editing, hopefully the gene editing will be smart enough to outpace the pathogen, because at the moment the war isn't really between the plant and the pathogen, it's between us and the pathogen in monocultures. But it's a very good point.
Yeah, it sounds it's a big paradox that you have to strike this balance between intervention and letting things run their natural course. And you're stuck between, on one end, homogeneity being your enemy and the need to edit genes. So how is it going to be a case by case? Is there a general rule about setting the cursor in the right place that you see coming, or?
No, I think it's incredibly complicated. So in human medicine, we have one host, us. In crop disease, we have 176 crops that feed the world. All the interactions between the crops and their pathogens differs. So take the potato, for example. You won't believe it, but the potato has 253 different pathogens. So every interaction between the host and the pathogen is stunningly complex. So it adds another layer, really. So I don't think there'll be a universal fix at all.
How much do we know about fungus? Because every time we go deeper in the ocean, we find new species. Every time we go to new parts of the Amazon forest, we find new species. Like, how much do we know about this world right now?
Well, I think we only know about 5% of the whole population of fungi in the world. So I think this is a huge resource when you just look at the things you can get from fungi in terms of new drugs, in terms of man-made polymers, well, not man-made, fungal-made polymers that we're now using for as alternatives to plastic. So it's an incredibly exciting time. It's just that people aren't really aware of the fungal kingdom. They think about plants and animals and bacteria, but not much about fungi. So that's my mission.
How easy is it to get some data about this? We saw in the previous talk that there were like networks of sensors being deployed to monitor the environment. Like, how do you have technologies that allow you to monitor?
Yes, we have technologies within crop plants, and we can monitor. But however much data we get, when we compare the simulated models with the real data, we've not got beyond a correlation of about 60 to 70%. But that's doing well, because it used to be about 20%. So we're getting there, but we need much more data.
There's a lot of data collection that needs to happen.
Thank you for your talk. It was really insightful. I had a question about, you mentioned kind of the sometimes unwillingness of these ag businesses to kind of share their fungicides. Do you see any world in which, as kind of this problem gets worse, there's more of a potential for academics like yourself to collaborate with some of these businesses on the kinds of research you're describing? Because I know I'm from Texas, a lot of my friends go into the agriculture industry. And I'm sure they'd be interested in subjects like this, but do you see any world in which that kind of collaboration is feasible?
Yes, so before I went to Exeter, I was 20 years in Oxford, and that seems to be quite a magnet for the companies to come to. And all of my PhD students were sponsored by industry. The problem with that was that they gave a small amount of stipend to the student, a small amount to the lab, but they wanted everything. And for the student's career, that wasn't really exactly what we needed. So I think it's different looking at antifungals in the mammalian world because most of those are small startup businesses with some very exciting leads. But in the crop world, it's the big companies like Syngenta, Bayer, etc. And they are very nervous about giving out data because they're so competitive with each other. But at least they're going towards what Lisa suggested, which is biologics as well. So we are beginning to open up. I'd like to think over 25 years, data becomes much more accessible so that we can look, for example, at combinations of fungicides to try and lower the rate of disease resistance emergence. It's a good point.
Hello, Miriam O'Doyle, Associate Professor of Chemistry. My question is, how much of a role does chemistry still have to play in this area, and how much are we moving towards gene editing? And tying in with that, you mentioned the delicate balance that has to be kept between different areas. The EU is just discussing outlawing PFAS, fluorinated compounds, but fluorinated compounds are incredibly good antifungal compounds. Does that have to be a discussion we can focus on in the future as well, or has that bus already left?
So chemistry is absolutely essential. And I'll tell you a little story. So we decided that if there were single target site antifungals, what we'd try and do is take the genome and design something that could target 2 or 3 different fungal processes. And we couldn't have done it without the organic chemists. So we synthesized a long lipophilic chain that would go into the mitochondria and put a sulfonium molecule on the top. And it's unbelievably antifungal. And that has been patented. And what we hoped was that the Bill and Melinda Gates might take that so it would be accessible for everybody. Unfortunately, they didn't. And now it's gone into a private company. So it's hugely disappointing to do something that you think is logical and then to lose it. But the chemists were extraordinary in telling us what we could and couldn't do without us really quite understanding what they were doing.
I'd like to take 2 more questions, so please, we have 1 minute and a half remaining. Let's be quick.
Sarah, Scott Phillips of Vault Adventures. Just back to the problem of complexity of the fungal biology. Do you think that, given that we now have quantum computing on its way and AI, and in fact quantum AI also becoming a thing, and powering simulations in future, do you think that computer simulations have a role now or in the future in this field?
Absolutely. Essential role. And in fact, what we've tried to do is we've run our mechanistic models, and then we've tried to also do them via AI. And those are being done at the moment in the Met Office in Exeter, because it's completely beyond my mathematical ability.
We have one last question there. Thank you very much.
Hi, thank you for your talk. So I started right after college. I did an internship in a biodiversity CGIAR sort of NGO, and I looked into bean and banana variety in Uganda. So I learned that, for example, Uganda was the biggest producer of banana in the world and biggest supplier. And listening to your outlook of, we are going to need monocultures to supply the amount of food that globally we consume. And the fact of my experience and thinking about things like in Italy, there's 400 different types of, for example, apple that are cultivated. Is there going to be a sort of compromise in going into monocultures where we see humans just getting less nutrients in their diets because we have to move to monocultures and we will lose varieties of food?
Well, I think if we could embrace gene editing, we could actually boost the quality of, say, nutrients within some of these crops. Bananas are an interesting one because the economy of India will probably be banana-based in the future because of climate change. There is a nightmare there because bananas, when you cut them in half and you see those 4 tiny dots, that's the vestigial end of their sexual breeding. So bananas are clonal. And we all eat Cavendish, and it's just succumbed to a new fusarium species. So we're going to have to be quite clever in the calories that we choose to plant. But it's a good question.
Thank you very much. That's a wrap up for today. Please give a warm round of applause to Sarah Jane Gurr.